Thank you very much for the opportunity to talk here today. And I have to say the presentations that we heard so far are really resonating with me. I might be referring to some of them during the talk. I am a little later on, so a few of the concepts that have already been mentioned, I will probably just glaze over. I want to talk today about some of these things that are going on at CHDI. And CHDI is a non-profit foundation. We have one mission only that's on the screen right now, which is to rapidly develop therapeutics that slow the progression of Huntington's disease. And I always tell everybody that we are living the dream of every pharma executive because we are an entirely virtual company. Everything we do is virtual. We only do wet work, so to speak, through third parties. And we are a science management organization, which basically means we do not just provide grants for the research in Huntington's disease, we actively help design and manage this work. And in that spirit is what I'm talking about today. And before I go any further, there's a lot of people at CHDI and outside that I need to thank. I wanted to make sure that the one group of people that don't get forgotten with the thank you are the study participants, because all the studies that I'm going to talk about have been based on very generous donations of samples and time and commitment from the participants to the study. So I cannot emphasize enough how grateful we are to those participants. And I'm going to tell you a little bit today about Huntington's disease. If I'm carrying Cole's the newscastle, just yell at me and I'll skip that part. I want to talk mostly about our clinical studies and our sample use in those studies. And because we're all cooped up, I did put some recent pictures from my garden into the slides where there was a little space to share some of the joy if I don't have an artificial California background to share. So I want to first mention what an amazing time we live on. And no, I am not talking about Corona. We did that during the panel. I'm actually talking about counting our blessings here. We really live in a time where fairy tales have come true. And if you just think of any of these or many other things you could list here, where the carriages move without ox or horse, where we can fly over the 7 mountains and are in the other kingdom long before the next sunrise, or we speak to whomever we miss most, even if they are far, far away, and our magic mirror shows us whatever we ask to see. So those are the things where really the question is, what is next on the list? And next on the list is that one thing where you give that drop of your blood to the wizard and he'll brew the stew that lifts the curse. And that's really what I'm going to talk about today, the importance of biosamples. Biosamples are amazing in the power that they bring. And I'm going to demonstrate this, of course, on Huntington's disease. Before I go there, I want to tell you a little bit about Huntington's disease. The reason we are working on Huntington's disease is because it is a devastating disease. It is deadly. It's a neurodegenerative disease. It is a genetic disease. It is autosomal dominant inherited. And it is considered a motion disorder, even though it actually has all the typical symptoms that make it described sometimes as having Parkinson's disease, Alzheimer's disease, and MS. So it is a horrible disease. The onset of the disease is generally midlife, even though there is juvenile Huntington's disease. And the diagnosis is typically given when the motion symptoms, the motor symptoms are manifest, but also nowadays on the gene expansion test, which is a gene test. It is in the larger group of repeat expansion disorders, The gene is Huntington. You will sometimes see in my slides the abbreviation HTT. And the mutation is a CAG repeat expansion in exon 1, which is of the Huntington gene, which is the fully penetrant version of the disease. The number of CAG repeats is varying, and I will come back to that later a little bit more. The amazing thing is that this gene has been discovered 25 years ago, and the mechanism of the disease is still not understood, even so we know already for a long time what the gene is. There are good information that points towards a toxic gain of function, possibly of the protein. There are also some people who still think it could be the RNA. Both have very good arguments on their side. You never know, in the end, they all may be right. The point being, we still don't know. And I told you before that I am going to talk a lot about biosamples. That is because my function at CHDI is actually, I am the clinical science director of biofluids. And I took this position because I was going to be in charge of the scientific strategy and execution of the biosample collections and distributions and uses, with a large emphasis on our biomarker work, which you will see later. And I just want to remind you where the use of biosamples actually started and how this developed. The classic way of biosample use is, of course, diagnostics and pathology. You can't do any pathology without actually having some sort of body or body parts or body samples in front of you. So this is a direct analysis of the individual sample, typically for the individual. And of course, you have what's already ongoing for a really long time, the targeted studies in the clinic, where you have a sample analysis from a larger group of participants, and you analyze the samples by a specific study plan. And you also have collections of samples that are reference samples, where you basically have already an idea what you're looking for, and you keep these samples to always be able to come back to these samples and put everything else you measure in a context. Right now, I'm mainly talking about the biomedical research sample collections, biomarkers, mechanism of action, and of course also still specific studies, which are really collection-based. So collections are a major part of what I am dealing with. And I will tell you right now a little bit about the impact of biosamples in Huntington's disease research that started these collections that I will later talk about, and that really show how justified it is to do this very important work. So in 1983, from the samples of 2 extensive family pedigrees, it was found already that there is a genetic marker for Huntington's disease. So these were some, if you count this, some 64 samples. Of course, there were other samples around that actually enabled this strategy. It was a relatively small number of samples, but interestingly enough, these initial samples were already DNA from cell lines that had been produced from the scientists who had really the vision into the future to not only create a blood sample that was frozen, but create already cell lines so that they had broad ability for future use. Based on this, the gene was actually discovered in 1993, and in 2019, we actually published a Cell paper where we showed that there are genetic modifiers for the disease. And to show you the difference in the scale, our paper that we just published last year was based on around 9,000 samples that were from a set of 1, 2, 3, 4, 5 GWAS that have been set up right now to still increase the number of samples. Interestingly enough, we are again back to the same sample type, which is DNA from cell lines that has been used for this GWAS to find the modifier genes. And I will mention a little bit later what actually I'm talking about when I say modifier genes. interestingly enough, for Huntington's disease, because the gene is known, there are already ongoing clinical studies. One key study has been published in 2018, and that study was based on CSF, because the strategy for clinical studies currently in Huntington's disease is based on our knowledge. And I mentioned earlier on that we do not very well understand the mechanism of action of the disease, but we do understand that it is the expansion of in the Huntington gene. So naturally, the strategy for these studies is right now in all the early studies the version of the modified Huntington gene. Accordingly, the samples that have been key to the success of the first study were CSF, where the amount of the expression of the modified gene has actually been measured, and it has been shown that the suppression of the suppression of the expression was successful. So this is just a quick run through on the biosamples and how important they are in Huntington's disease research. I want to put this into the context of biosamples in general. I will focus this a little bit on neurodegenerative diseases, but you can expand that to a lot of other diseases. Huntington's disease, in contrast to, in particular, Alzheimer's and Parkinson, but also partially still MS and a lot of other diseases in the neurodegeneration field, has the, if you want, advantage that I just showed in the last slide extensively. We do know what the gene is. So we can stratify our patients by who is going to get the disease and who is not going to get the disease. We know that. We also know what the disease is. We have a fairly good understanding of when the disease onset will be, because it is known by now and very well shown that there is a correlation between the onset of the disease and the extent of the expansion in the Huntington gene. And we call this a CAP score, and out of that we can actually determine roughly when the onset of the disease is expected for an individual. However, in the graph that I'm showing here, when I'm talking about the prognosis, I am also showing that, if you cannot possibly read this, on the x-axis, this is the age of the CAG repeat length, and on the y-axis is the age of onset for the person. And you see that red line, which is what this CAP score actually predicts. What you see also is that there is a very wide spread, and I will come back to that later. This spread is basically the difference in age on onset for the individual versus what you expect from the CAG expansion that you see, that you measure in their cells. And that's where our natural history data and our populations come in. And I'm showing here a Manhattan plot from the GWAS that I was talking about, because the variation in onset in CAG, in the disease, and the variation in the CAG repeat seems to be dependent on other factors. And those are the modifier genes that I was referring to that have been described in the GWAS findings I mentioned earlier. Now, for a lot of neurodegenerative diseases, these are luxuries that these diseases don't yet have. And that affects, obviously, the strategies for the sample collections. Now, moving forward, I'm only going to talk about what CHDI is doing, and CHDI is exclusively working for Huntington's disease. But I want to assure you that we are thinking about the broader context, and that the community in the neurodegenerative disease research is looking at these sorts of studies and learning and neurodegenerative diseases, but at the same time that will be applicable fairly broadly. So having said this, I want to talk a little bit about the clinical studies. And I want to start with a list of some of our current platform and sample collection studies. CHDI is actually financing a lot of studies. And I want to draw your attention first to the orange line, to Enroll-HD. Enroll-HD is our platform study. It is a natural history study, and yes, this is not a typo. We have more than 20,000 participants in the study. It's a longitudinal study. You see here on a timeline that it started in 2012, and it actually overlapped with predecessing studies. I listed here only registry, which was a mainly European study that is now being rolled into Enroll, but that adds data and samples to the study even more longitudinally. And it's only possible to create this sort of study because we understand our population really well. You see also that we started adding more studies. We have a study, the yellow line, which is called HD Clarity, which is a collection of CSF and plasma. And this is going to be a longitudinal 5-year study for about 1,200 participants, where we started about 2 years ago collecting samples for understanding CSF. That is based on the needs in the clinic, because CHDI is trying with all of their work to really bridge that valley of death that we see quite often between the insights that are being gained, in particular in academia, and bringing this into clinical studies, bringing this into something that can be moved forward to becoming treatments for patients, because that's our main focus. We want to get these treatments, and whatever is needed to get there is where we try to support the community. Accordingly, we have HD Clarity, which is solely dedicated for collecting these samples, and I will talk about it a little bit more in a minute, to support the research community. Any comer who wants to have insights and who needs samples for that can come to us and ask for these samples. We also have an independent plasma studies just started. It's called Enroll Plasma, the purple line. And I will talk a little bit about Origin HD, which is our semen blood study, which is a little different in the sense that it is actually a hypothesis forming and testing study. Let me give you a little bit of an overview over Enroll HD. As I said, it's our huge platform study that's already ongoing for a long time. And you see here that as of January this year, we had 19,521 active participants. We have a few participants who over the years either dropped out or passed away, whereby our retention rate is amazing, actually. So that we have a total right now of over 20,000 participants in the study. We have almost 180 clinical sites worldwide, with about 20 nations participating. And for the samples in this study, we are collecting blood, we are analyzing the CAG repeat for the participants. We had earlier today a conversation or a presentation that mentioned, should we tell a participant or a patient about their disease? For Huntington's disease, there is no treatment yet. And so when we do genetic testing, we actually are making sure that nobody who doesn't want to know their status of the gene is confronted with that sort of information. That's just as a side note, a follow up on the conversation or presentation earlier today. So we are paying great attention to the protection of our study participants, but at the same time, we are collecting the blood samples for creating cell lines, which lately we stopped because we have a lot of cell lines, but we still have the blood, we have the DNA, we collect PBMCs in this study. And we have a large data collection. I always say that clinical samples without the data is actually nothing but a collection of very expensive to store frozen groups. And in that sense, the data that is coming with our samples that is very well describing our population is really what makes these samples so valuable. The next study I wanted to mention a little bit about is HD Clarity, which again is our CSF and plasma study. We also collect a little bit of serum. We have about 1,200 participants, and it is a longitudinal study where I cannot emphasize enough the heroic and really dedication from the Huntington's disease community, the patients, and that was mentioned earlier today too, the patients, their families, that community are the drivers in the end of everything we do, not only because we feel their pain, but also because they are so engaged, they are so giving, and a CSF collection is not considered a large procedure, but it's certainly not just a finger prick. And it takes a little bit of courage to do this and to do this repeatedly. And so we have this dedication from the patients who come back and give us these samples. So I will talk a little later on some more about that. Besides this commitment that we are getting from the patients, from their engagement that we owe to them now, also to make the best research use, we are working hard to make these samples have the highest impact possible on the research in the Huntington's disease field. Before I go on and talk a little bit more about that, I also want to make you aware that we have a lot more studies. I mentioned earlier that my function at CHDI are the biosamples to be collected. However, we are very well aware and have very much work going on with other biomarkers and not just the wet biomarkers. We have imaging studies that are coming up right now that are of course correlated with sample collections. We have outcome studies that are very important, where we have in particular also socioeconomic insights, where we work on very important rating scales. Those are right now our best biomarkers that we have for neurodegenerative diseases. I mentioned the plasma study already. I'm going to talk a little bit about origin, and we have more to come. I mentioned during the panel discussion today that we have a virtual study that we are bringing up right now, and that we have more studies in the planning. We're really trying to fulfill the needs of the research community and make sure that we can deliver what is needed today rather than following behind. It's not always easy. Sometimes the success of what we're doing is catching up with us and is catching us on a place where we have not yet the infrastructure, but a lot of those subjects have been discussed today, and I don't want to right now go too deep into that. I want to talk a little bit more about the sample use. I mentioned already how important to us the commitment of these samples to the right kind of use is, in particular since we are collecting most of these samples with a perspective of not knowing exactly how. But we have, of course, an overall strategy of what these samples are supposed to do. And they are supposed to fulfill two major needs. One is a better understanding of Huntington's disease. It is quite a frustration, actually, that we do not have a really deep understanding of the mechanism of action. And again, I'm not going to talk about it, but CHDI also has a large preclinical team that is working on the translational and preclinical research and managing research that is really driving towards gaining more insights, getting more ways of understanding the disease so that it can get more treatment approaches. But also we want to, of course, directly support the development of new treatments. And so for understanding Huntington's disease, the way we try to optimize the scientific gain from our samples is by providing things like reproducible assays. I do spend a lot of time using my history in assay development and assay understanding, assay technology to help build and test and solidify reproducible assays. At the same time, I will later talk a little bit about providing seminal data where we can centrally use our samples to provide data instead of samples for questions that everybody needs to have answered and that we don't need to have repeated in a lot of small studies if we can do that one decisive study. And that is basically one of those things that help us go towards understanding of HD. At the same time, for the support of the development of treatments, we do a few things the same. We analyze samples, we gather data for modeling support, getting models, we had a lot of conversations today about digital medicine, about using the data, gaining the data, understanding the data. Now the data is coming from somewhere. And in my mind, actually, I told you already that a sample without data is nothing but a blob of goop that's really expensive to store. There is another aspect to a sample to me. To me, a sample is actually nothing but a little box, black box of really precious data that has not yet been extracted. And so we do go and we do extract data from our samples that we help really for building these models, and that we also use for helping analyze the data from clinical studies, prepare clinical studies, plan clinical studies. And that's where we again provide samples and data to support these activities. Having said this, so for supporting and accelerating the development of these treatments, for these 2 points, we go about our samples in a number of really key ways. One is, of course, we use the unbiased approaches. So I am working, of course, on a number of omics projects where we are trying to do the hypothesis forming research to find new insights. We do this sometimes or most often in collaboration with external groups that are interested and who we support. If nobody is doing it and as is needed, we may be doing it on ourselves, on our own. Also, in particular in the biomarkers field, there are a lot of hypotheses that need to be tested. They need to be understood. Can we make this into a clinical biomarker, or should we put the resources of the community somewhere else? So we go and we do test specific biomarker hypotheses. I talked already about the support for modeling, and the majority of our samples is actually being used on the outside of CHDI. So we have a process where researchers can request our samples and our data in order to support their own research. And to just give you an idea for HD clarity, basically, we are trying to use these samples in, of course, I mentioned already the HD lowering trials, and I showed you in an initial slide that the first trials used CSF in order to support the Huntington lowering studies. So of course, we use our HD clarity samples to support that sort of activity. We are also, I talked about the hypothesis testing. We are testing markers for neurodegeneration pathways to see whether we can find out more information, which neurodegeneration pathways are involved in Huntington's disease specifically, and how are they affected. And we are testing metabolic pathways, we are testing signaling pathways, and also we are trying to use new ways of analyzing samples and new samples. So of course we are following with the extracellular vesicles and any developments where, for example, for extracellular vesicles, we haven't yet set up our own research. We probably are going to work through others because there is a lot of interest in the field where we just have to help with some coordination, where we just have to make our contributions where we can, rather than having to do everything for everybody. And for HD clarity, I mentioned in the first point that we are working on some of those seminal data, and just wanted to give you some idea of what that looks like. So in clarity, CSF, as I said, is not easy to give, is a very valuable and fairly rare resource. And so we don't want to have everybody come and measure the markers that are typical for the disease in order to establish their own baseline. And the only way we can avoid this is by providing robust data that actually help understand the disease and that help doing the planning for which you need data when you do your studies, when you analyze your data. So we actually do have, we do develop protocols. We have a process for sample delivery and analysis. I have a massive quality control effort going on. We do prepare data sets from that, and we distribute and analyze the data. We do our own analysis, but we also distribute it for anybody who wants to do their analysis their way. And right now we are doing this with the CSF and plasma from HD clarity. And for example, of the CSF right now, of the planned number of samples, I have data for about 37% of these data. of these samples, and the crisis did hold that back. I hope that the labs will soon get back opened and that we will get more data, but the safety of the people in the lab, quite frankly, is sometimes just way more important than driving the deadlines for the data, and so we will get this when we're ready. And I want to talk a little bit about the study that I mentioned already a few times, which is Origin HD. And Origin HD is following up on a very important aspect of repeat disorders in general and Huntington's disease in specifics that is already fairly long known. And that's what's called anticipation. Anticipation basically means that I told you earlier that the disease onset is somewhat correlated with the number of repeats. Long before the gene was known, it was already known that quite often when the disease is passed on from one generation to the next, the disease gets an earlier onset and sometimes also a more aggressive development of the disease in the younger generation. That's called anticipation. And this graph here represents this, where the black bars are basically the age of onset for on the x-axis for the parents. And you see that for the children, the white bars are shifted to the left because their age of onset in general is younger. Now that we know that the disease is an expansion, we also know that the younger generation generally has a longer CHG repeat. So we know already that anticipation has something to do with the expansion of the repeat. And the Origin HD study is different from the Clarity and Enroll studies, which are platform studies, in the sense that we are following right now here a hypothesis. We are testing it and we're trying to form more hypotheses to understand the phenomenon of anticipation and put it also in the context of what I'm going to tell you a little bit more about the disease and a little bit that we think we understand about the mechanism. Let me just mention to you that this is a study where we are collecting semen and whole blood, and we're doing this for about 1,000 participants as a cross-sectional study. So this is not a longitudinal study. We're going to do, hopefully within 2 years, the collection of these samples and gaining more insights as we are moving forward. Now, I mentioned already that the length of the CHG repeat changes between the generations, and that we know that being obviously underlying the phenomenon of anticipation. We also know by now what is called CHG instability, and that's what this graph shows here, which is that within a person, if I say your CHG repeat is 42, that may be so. However, it turns out that in some samples and in some cells, actually over the lifetime of a person, the number of CHG repeats changes. It seems to be more towards longer CHG repeats, and we are expecting right now that this change in CHG repeats is one of the underlying mechanisms of the onset of disease and probably also of the disease development over time. So with that being said, we are using for Origin really the information that we gain for germline instability. That's why we do the semen collection. We want to look at somatic instability. That's why we're doing the blood collection. And we want to look at the modifier gene variants, which we can get again from the DNA we are collecting and integrating the knowledge from the GWAS. We bring this together and we hope to really understand the mechanism of action of the disease much better. Of course, in the end, with the goal of finding new approaches for more treatments that could be developed for Huntington's disease. And our sample use, oh, okay. I see that my time is running out. So I will skip over a few slides here. I wanted to tell you a little bit about who's using our samples and for what. Let me just summarize that by saying everybody, industry, academia, CROs, everybody is very actively using our samples for all sorts of different research fields, as you see listed here. And we also are very strategic about the way we are distributing the samples. I will skip that as well. I am planning to publish on that hopefully in a few months if I get the time to write it up. And let me just summarize that we hope to really be a major contributor to driving the research field for Huntington's disease and hopefully with what CHDI is working. And I will be happy to take any questions.